Welcome to Unit 7. In this unit, we will be discussing what genetic testing can do for us today. And in this chapter, particularly, what would direct-to-consumer genetic testing really do for us? So let's take a look at our case concerning genetic risk assessment. Meet Neil. Neil is a young man who's at a dinner party, and people are talking about getting their genetic profiles. And one of his friends says, well, I went and got my genetic profile, and now I'm changing my lifestyle, so I will live longer. And his friend did this with what we call direct-to-consumer testing. It has, of course, and continues to generate a lot of controversy. Although it is not marketed as medical testing, it does come with disclaimers, but it's really impossible how individuals will respond to the results or react or really do anything with them. And however, because they are doing testing, they have to be certified by CLIA, the clinical lab. Of course, Neil's friends were talking about this direct-to-consumer testing, and there are both supportive and opposing arguments for these. Those that support it says the genetic information belongs to the individual, and if it's your genetic profile, it should belong to you. And the medical profession, therefore, shouldn't stand in the way of your obtaining that information because it's yours. Those who support this will claim that it's educational information as good or better than what you would get from the AMA. I kind of doubt that. But many do not offer, but some do offer genetic counseling once someone gets their results. However, what about the opposing arguments? Well, this might make people make medical decisions without the benefit of medical advice, and that means they're not really capable of interpreting this information in a proper manner, and that is true. This stuff is difficult, and people can overinterpret it or underinterpret it in some ways, if you will. So most of the tests also have, although the labs that are performing these things are certified, but the actual tests themselves have not been validated either for clinical validity or utility. So what use is it really? And the consumers will not understand what test limitations are. It's on the internet, you got the answers, therefore they believe it. Now, I know you guys don't do that, but a lot of my freshmen undergraduates do. And as a result, they get information like this, they're going to make unwise decisions. In other words, they might, I got to get into some crazy fad diet to lose the weight, or I need more expensive follow-up tests that they really don't need. And as a result, they may do things or not do things that's actually really bad for their health. But anyway, or good for it, as the case may be. There's really very little consumer guidance when it comes to having this done. So, part 2. Neil decides, well, I think I'm going to go do this anyway. So he contacts one of the companies, and he receives a package from the testing company, as you see over here on the side. And of course, I couldn't resist putting a little picture of Neil on there. And what he gets is a plastic test tube. It's labeled with a barcode. It's got this funnel-like cap so he can spit into it. And then there's a plastic bag and a mailer to return it to the company. And as it tells in the little story, he thought, it's not that easy to spit onto this, but eventually he's able to do it and to send it back. So what's really being tested? Well, let's see this on the next slide. So it's really a genome-wide association testing for single nucleotide polymorphisms, or SNPs, okay? What they're using is rapid, low-cost microarrays. We've talked about these in previous chapters. And of course, they're made, just to remind you, they have specific oligonucleotides that have different polymorphisms on there. are attached to a glass chip, or in some cases actually a silica bead. And then these oligonucleotides are different alleles of a particular polymorphism. Okay, so they'll have slightly different sequences. Now, if a patient has an allele, his DNA will hybridize, remember this complementary base pairing, A with T, G with C, with the oligonucleotide. If the oligonucleotide and his DNA become single-stranded, which is easy enough to do when you heat it. You separate the two strands. You break the hydrogen bonds that are holding it together. Now, the nice thing about this is because you can test hundreds of thousands of SNPs, single nucleotide polymorphisms, simultaneously, and it's not the saliva, but the DNA from the epithelial cells that are in your saliva that is actually providing the DNA for the testing. This figure is from your textbook. It shows that you have oligonucleotides attached to the silica beads. The beads themselves are etched onto a solid support, in this case a glass slide. And then you have different patients, and of course their DNA has to be made single-stranded, in which case they will be able to base pair. If they're homozygous for the A, then they will base pair to it. Here's because they've got to have complementary. If they're heterozygous, they will base pair to one that has the A, the other one that has the T. And then if they're homozygous for the T, they only base pair with the T, okay, because they'll have the A. So this is showing you how they are able to screen for these polymorphisms. And of course they're really, really small, and so you can put literally hundreds of thousands of them on a single glass slide. And then a computer reads it so that you can see the kind of results we see on the next slide. And here's the next slide showing you the same thing. In this case, if we look over here on the lower right, you can see single nucleotide polymorphism, or once you have an A in there, another G, another T. And this is really what it looks like. And you can see all these different little beads, and they're fluorescing because you labeled the patient samples, so they fluoresce when they bind to one or the other. And the computer, of course, knows what each of these polymorphisms are, and it takes a look at it and interprets it for you. So Neil sent it in, and he gets his results. And what he finds is, like most people who have this done, his risks are a little higher or a little lower than the general population's risk. However, he does have, his paternal grandfather had Parkinson. And if you take a look for it, that is increased threefold for him. He does seem to have a gene that would increase his risk, and of course he also had a first-degree relative as well. All the other things, however, are either a little bit above or a little bit below, and this is typically what people get. The company is really just calculating the odds ratios, which we learned about in previous units. And this is the ratio of the disease in individuals with a particular single nucleotide polymorphism compared to those without it. Okay? Analytical validity is high with this kind of testing. If the SNP is there, it is detected. But, of course, human error, the sample mix-up, and everything can occur. Clinical validity is not very high. How useful is this? Well, risk assessment is determined in a specific patient sample, usually with one particular population, but there's different populations have different risk estimates. The disease risk is calculated inheritance of multiple SNPs typically. When we're talking about things that have been done in a larger study, each company will also use different sets of SNPs. So, you're comparing apples and oranges is what it's trying to say. One study showed significantly different disease risks from each of 5 different companies. Okay. And they, of course, don't take into account that disease risk is also affected by environmental factors. And then other genetic factors can contribute, but they're not tested for. Okay? So there are inherent problems with this. And clinical utility is very low. There are very few common disorders that have specific risk modification regimens. There's no data showing results of these direct-to-consumer testing make people eat less and exercise more. Some with a reduced risk might think, well, I can eat more and exercise less. So really, clinically, the utility of these is very low. It's just informational. And so I couldn't resist putting food and, of course, couch potato. But when I Googled couch potato to get a picture, I got to tell you this. I was they were having some pictures of men and women sitting there. But when I saw the cat, I just cracked up, and I had to put that there. I mean, notice the beer and the remote in the hand. But look at the face of that cat. That cat looks like she's really watching the television. So I love cats, don't get me wrong, but I just couldn't resist this. I hope you take it right. Okay, part 4. So now we're going to look at something that might be a little bit more interesting, metabolic and carrier risks. Okay, so Neil's test reveals he is a slow metabolizer of caffeine. He knows this already because if he drinks coffee at bedtime, he can't go to sleep. I have the same sort of thing. If I have a pot of tea with dinner, to end with dinner, I couldn't figure out why I couldn't go sleep every time I went out to dinner someplace nice. And finally I figured it out. But anyway, what we do know, and we'll be talking about this in Chapter 16, is that the absorption, metabolism, and excretions of different chemicals are influenced by specific genes. And pharmacogenetic SNP testing can actually help to choose medications with less side effects. But this is only useful if the patient and or the physician knows about it and understands the results. And as I said, we'll be talking about this in a little bit more detail in Chapter 16. And Neil finds out he's a carrier for cystic fibrosis. Now, this is, remember, these tests do have high analytical validity, so it's probably correct. However, he should have further testing to confirm it because he want to make sure it is a pathogenic, if you will, or pathologic SNP. And they can miss certain SNPs, and this may not be the better test for screening for cystic fibrosis. Tay-Sachs, for example, you'd rather do the blood testing than the SNP testing because there's so many different mutations that can lead to this. And that's partly the true case with cystic fibrosis. But let's face it, it's also recreational genomics. It predicted his eye color correctly and whether he can detect certain odors and not. And let's face it, this is kind of fun stuff, and so people enjoy doing that. We use this in genetics labs for undergraduate students. They get a chance to see whether they can taste certain things or smell certain things. And some of them can, and some of them can't. So some of the stuff is kind of fun to learn about. But then we get to the warning message. And Neil reads this message that warning him that he's about to view results that can be kind of scary. And these are his risk assessments for cancer and Alzheimer's. He knows there's no family history in his family for Alzheimer's, but he does have a maternal aunt that died of breast cancer. So he eventually goes in to take a look at these, and he finds he's negative for BRCA1 and BRCA2, and he has the same risk for Alzheimer's as the general population. So that's not so bad. Typically, this is what you find out. The direct-to-consumer genetic testing rarely reveals any significant increase or decrease in disease risk. And the direct-to-consumer targeted gene tests, such as BRCA1 and BRCA2, are not as comprehensive as the ones that would be done if you're going to a genetics clinic and everything, because they'll just do the major polymorphisms, so they'll miss mutations, or they'll find mutations, but these are mutations that don't increase your risk of developing it. So what we call a non-pathologic mutation. Now, Alzheimer's, of course, is a common cause of progressive dementia in the general population. There are some specific alleles for the APOE or ApoE genes. APO is actually a lipoprotein that carries cholesterol, and it has three alleles. Epsilon 2, 3, and 4, which can be identified by the SNP testings. We find that people of European ancestry, heterozygous for epsilon 4, have a twofold higher risk, but if they are homozygous for epsilon 4, they have 11-fold higher risk. We don't know why this increases the risk, and even if you do find out about it, there's no way to decrease your risk as at this point. So knowing an increased risk may or may not cause psychological harm, and that's one of the major controversies, of course, with this testing. Again, people who are not don't really understand the genetic basis for these tests can be very depressed by finding out that, oh, this is what's going to happen to me, etc., etc., and they don't understand that risk does not mean that you will definitely get a disorder. So, genetic screening for disease risk is supposed to detect risk in asymptomatic individuals. This is one of the promises of genetics in medical practice. We hope it will offer intervention, prevent disease, or at least serious complications. There are some advantages to genetic testing. It can be done at any time and only needs to be done once, if you think about it. Once you know your genetics, well, they don't change. And it's non-invasive. You can have a blood sample, or you can spit into a tube, or take a little swab of your cheeks. Genetic testing pitfalls, it has limited predictive value. And because of things like penetrance and disease variability, we really don't know, just because you have an increased risk, whether you're, as I've said on a previous slide or two, whether you're going to get the disease. And even if you do, well, what clinical utility was there? There wasn't anything you could do to decrease the risk in some cases. So there are still some pitfalls with this. Well, there are two types of genetic screening tests, and I do expect you to know these. Number one are pre-symptomatic tests. Now, these tests are performed for disorders that display age dependence, and they have complete penetrance. So we know the gene. We know that if you have a particular polymorphism, that means you will get the disorder. But you can't predict the time or the severity of the disorder. However, this prepares you for the symptoms and allows you to do some family planning. In contrast, like this directed to consumer testing, those are all predispositional tests. All they're doing is estimating a risk. They're not telling you that you will get a particular disease. They can't predict whether you'll get it or not, and one of the main reasons they can't is because they are applying this to common multifactorial traits, as opposed to the pre-symptomatic, like you see here with a young woman who has Huntington's disease, and of course they have muscle contraction, weak muscle wasting and everything with that. And that can be, that is something that a lot of times a parent does not know they have until after they've had children, because it usually strikes in their 40s. And then, of course, they're concerned about passing it on to their kids. So pre-symptomatic tests have not only high analytical validity, but definitely clinical validity. So, can you reduce disease risk? You can, depending on what you're testing for. For example, with certain familial cancers, surveillance is important. If you know you have an increased risk, you may be more or less vigilant depending on the test result. You can go ahead and do preventive surgery, such as done with colon cancer and breast cancer. Colon cancer, they can remove the polyps early. You can modify your diet and exercise, and that does help with a lot of things. And a lifestyle modification, you can quit smoking and drinking if positive. Some people, unfortunately, might start smoking and drinking if they have a negative trait. Well, hopefully not. But that's the whole thing, because we cannot predict, and they're not getting medical advice along with their genetic results. So these are here. I think this direct-to-consumer testing is here to stay, but I would not, on a professional basis, necessarily recommend it. I'm thinking of doing it because I think it would be fun to be able to use my own example as to how I felt about reading some of this, even though I know a lot of it is kind of silly. But I think I might do that for the next group, and I'm also going to be kind of reworking this test into a 2-semester pathophys course that we're going to start offering for the master's course, not for the DNP course, in the future. So keep in touch, and I'll let you know what my results are. Meanwhile, go ahead and go to chapter 15.